Have you ever peered into the intricate universe residing within your own skull? I mean, really wondered what's actually happening at the most fundamental level. How do, stray thoughts solidify into brilliant ideas? How do fleeting sensations become these deeply felt emotions? Every memory, every movement, every single decision you make, it's all orchestrated by this, well, impossibly complex dynamic communication system. Today, we're embarking on a truly fascinating deep dive. We're plunging right into the realm of neurotransmission within the central nervous system, exploring the brain's secret language, how its billions of cells communicate, and maybe even more vitally, how powerful medicines interact with this really delicate, intricate balance.
Yeah, and our mission today is, well, ambitious. We want to take a stack of foundational pharmacology texts. These are sources brimming with highly technical information, and really distill them into a comprehensive, you could say, mastery level understanding. By the end of this deep dive, you'll not only grasp how the CNS truly functions, but also precisely why this knowledge is so indispensable. Indispensable for treating a vast array of conditions, everything from the subtle anxieties of daily life, right through to the profound challenges of Parkinson's disease. We're going to unpack every concept, every single nugget of insight. The aim is to ensure you'll be speaking the language of brain chemistry, like the elite professionals in this field.
And rest assured, this isn't going to be some dry lecture. Think of it more like we're decoding the brain's symphony, piece by astonishing piece. We'll start with the fundamental building blocks, the cells. Then we'll uncover the electrical sparks and the chemical whispers that connect them. We'll meet the messengers themselves, the neurotransmitters, in all their, frankly, astonishing diversity. And finally, we'll journey into the cutting-edge world of how we harness this incredibly precise knowledge for groundbreaking drug development. And this is where it gets really interesting. 1. The brain's orchestration. An overview of CNS function and pharmacology. Okay, let's begin with a broad, high-level view. The central nervous system. We often think of the brain like a super powerful computer, right? But the research, it paints a more vibrant picture. It's described as a complex assembly of interacting cells that orchestrate many of life's activities in a dynamic fashion, mainly through something called neurotransmission. When we say dynamic, what does that truly mean for how our brains operate day to day?
That's a great starting point. When we describe the CNS as dynamic, we're really emphasizing that it's anything but a static, sort of hardwired machine. It's a system in constant, ceaseless activity, always adapting, learning, responding. Imagine a vibrant, flowing conversation happening across billions of cellular networks. These networks are constantly rerouting, strengthening connections, maybe weakening others, all based on experience. This incredible dynamism is what allows us to learn new languages, or form complex memories, or react in just a split second to something sudden in our environment. It's not just a collection of switches, it's like a living, evolving orchestra.
That makes sense. And because the CNS has such widespread influence, governing everything from our heartbeat to our highest thoughts, it makes perfect sense that drugs acting on it are, well, invaluable for a whole plethora of conditions. We're not just talking about mental health here, are we?
Precisely. No, not at all. While centrally acting drugs are absolutely foundational for managing conditions like anxiety, depression, mania, schizophrenia, their impact stretches far, far beyond psychiatry. They're critical tools in treating really diverse pathophysiological conditions, things like chronic pain, fever, movement disorders like Parkinson's, and even common issues like insomnia, eating disorders, nausea, vomiting, debilitating migraines. When you think about the sheer spectrum of human experience and suffering that's touched by these medications, their importance is just immense. But it's also crucial to acknowledge the potent dual nature of these agents. The recreational use of some CNS-acting drugs can, unfortunately, lead to significant physical dependence and profound societal impacts. It really illustrates the immense responsibility that comes with understanding and developing them.
Okay, so given how vital and widespread these drugs are, you might think developing them would be a well-trodden path. Yet the research consistently highlights these extraordinary scientific challenges in finding targets and developing drug molecules for the CNS. It's even stated that a complete understanding of the functions of the human brain remains in its infancy. Why is creating effective brain-targeted therapy so incredibly difficult?
Well, it really boils down to the inherent complexity and interconnectedness. It's fundamental. Unlike, say, a bacterial infection where you might target a single specific pathogen, the brain is this highly integrated network. A CNS active drug often acts at multiple sites, and those actions can sometimes have, well, disparate or even opposing effects. It's a bit like trying to fine-tune a vast interconnected orchestral system where adjusting just one instrument subtly changes the sound of many others, often in ways you just can't predict. And furthermore, many CNS disorders aren't confined to a single faulty component or pathway. They likely involve multiple brain regions and interconnected pathways all at the same time. This sort of distributed nature really frustrates efforts that focus on a single therapeutic agent, because addressing one part of the problem might not resolve the underlying systemic issue at all.
So it's like trying to fix a really complex network problem with just a single localized patch, and then having that patch ripple out in unexpected ways. That's a powerful analogy for the challenge. Yet despite these hurdles, CNS pharmacology pursues these two broad, often overlapping goals. One is to develop and use drugs as biochemical probes to both elucidate and manipulate the normal CNS. And the second, of course, is to develop drugs to correct pathophysiological changes in the abnormal CNS.
Exactly. And what's truly fascinating here is how these two goals are intertwined. It's like a continuous feedback loop. The very tools we create to heal the drugs, they also become our most powerful instruments for discovery. By observing how a specific drug affects normal brain function, we gain these profound insights into its underlying healthy mechanisms. And this understanding then directly guides our efforts to fix what's broken in disease states. It's this kind of symbiotic relationship between research and therapy, constantly advancing our knowledge base.
And thankfully, we are seeing real progress in this incredibly challenging field, aren't we? Fueled by modern advances. The research points to breakthroughs across molecular biology, neurophysiology, structural biology, epigenetics, biomarkers, immunology, a whole array of specialized fields. These aren't just academic buzzwords, are they?
Not at all. No, these are the absolute bedrock for understanding the brain at an unprecedented, almost atomic level of detail. Consider techniques like X-ray crystallography and cryo-electron microscopy, or cryo-EM. These aren't just theoretical tools. They allow us to literally solve the precise 3D structures of critical receptors and proteins to an incredibly high degree of accuracy. Think of it like getting the exact detailed blueprint of a complex biological lock. This structural insight then feeds into sophisticated computer-aided molecular dynamics simulations and modeling. This allows us to virtually test how neurotransmitters and potential drug molecules might fit into and affect these signaling proteins before we even synthesize them in the lab. And as more of these atomic level structures of receptors, transporters, ion channels are elucidated, these structural biology techniques will become even more prevalent, even more crucial in drug discovery and development. It's truly revolutionary. It's almost like having an engineering manual for the brain's tiniest components, enabling us to design the perfect custom-fit key for specific biological locks. 2, the brain's architecture, cellular organization of the CNS.
Okay, with that incredible big picture in mind, let's zoom in a bit. Let's look at the brain's fundamental architecture. At its core are the neurons, right? The brain's primary communicators. They're described as highly polarized signaling cells. What makes them so central to everything our brain does? And how do scientists even begin to classify their, well, astounding diversity?
Right. Neurons are indeed the fundamental units of communication. They're uniquely adapted to transmit both electrical and chemical signals. Their astonishing diversity means they're classified in all sorts of ways. By their specific function, are they sensory neurons detecting input, motor neurons driving action, or interneurons connecting other neurons? By their precise location within the brain, by their unique morphology, their physical shape, which can be incredibly elaborate, by the specific neurotransmitters they produce and release, and even by the specific class or classes of receptors they express on their surface. And crucial to their role as communicators is their electrical activity. These aren't passive cells, they are electrically active. That means they express a sophisticated array of ion channels and transport proteins that enable them to generate and conduct these rapid nerve impulses or action potentials. These potentials ultimately trigger the release of their chemical messages. Furthermore, neurons are cytologically highly active secretory cells. They're packed with the machinery large nuclei, extensive endoplasmic reticulum, specialized Golgi complexes for constantly synthesizing and packaging these chemical messengers.
And the actual communication points, those critical junctions between neurons, they're called synapses. These are the sites of interneuronal communication. The research functionally compares them to junctions in the peripheral nerve system, but with a much more intricate setup. It involves this array of specific proteins presumed to be the active zone for transmitter release and response. And within these synapses, these tiny synaptic vesicles, just 50 to 150 nanometers in size, they're absolutely packed with specialized proteins. Proteins necessary for neurotransmitter storage, vesicle docking, secretion, and then reaccumulation. This really paints a picture of an incredibly intricate and tightly choreographed dance, doesn't it?
It truly is an intricate dance, a micro ballet, you could say. The precise release of these neurotransmitters from the presynaptic neuron and their subsequent recognition and action on specific receptors of the neighboring postsynaptic cell, that's the fundamental mechanism. It underlies the ability of these specialized cells to communicate with each other. And this continuous, precise communication dictates all our complex physiological actions. It forms the very basis of our thoughts, emotions, and movements. Understanding this dance, this interaction, is really key to understanding the brain.
But neurons aren't alone in this grand orchestration. Our sources introduce us to the non-neuronal cells, often called glia collectively. These are the unsung heroes of the brain. They actually outnumber neurons in the CNS. Right. Traditionally, glia were thought to just play a supporting role, but that view has significantly changed, hasn't it?
Yes, dramatically so. While neuroglia absolutely perform vital support functions, holding neurons in place, supplying essential nutrients and oxygen, insulating signals, acting as the brain's immune system, newer studies have unequivocally shown something more. They've demonstrated that they may also be involved in some signaling processes. So they're far from passive bystanders. They're active participants, constantly modulating and protecting the neuronal environment. Within the macroglia, the most abundant are astrocytes. These star-shaped cells are intimately interposed between the blood vessels and the neurons. They actually surround individual compartments of synaptic complexes. Their roles are crucial. They provide metabolic support, they furnish energy intermediates to neurons, they anchor neurons to their blood supply, and they actively regulate the external environment by active removal of neurotransmitters and excess ions, basically maintaining the precise chemical balance needed for neurons to function properly.
And then there are the oligodendrolia. These are truly fascinating because they produce myelin, those multi-layer compacted membranes that electrically insulate segments of axons. This insulation is critical, right? It enables non-decremental propagation of action potentials, meaning the electrical signals can travel long distances without losing strength, much like insulation on an electrical wire ensures current flows efficiently. We also have ependymal cells, lining the spinal cord and ventricular system, involved in creating cerebrospinal fluid, and radioglia, which play a vital role during development, acting as neuroprogenitors and scaffolds for migrating neurons.
Exactly, and we absolutely cannot overlook the microglia. These are the specialized immune cells within the CNS. They act as the brain's resident macrophages to protect the neurons and are the mediators of immune response.
Yeah.
They're exquisitely sensitive to changes, responding rapidly to neuronal damage and inflammation. Crucially, the research shows that the balance between the numbers of microglia and astrocytes can determine whether there will be resulting cell damage or protection in situations of chronic neuroinflammation. So if we connect this to the bigger picture, these so-called support cells are far from secondary players. They are active, dynamic participants, constantly modulating and protecting the incredibly delicate neuronal environment. And increasingly, we see them as crucial players in both healthy brain function and in the progression of many disease states. Understanding their intricate roles is just as vital as understanding neurons themselves if we want to develop comprehensive therapies.
Speaking of protection, the brain has this extraordinary, highly selective bodyguard, the blood-brain barrier, or BBB. It's described as an important boundary separating the periphery from the CNS. And it's not just a simple wall, is it? It's composed of specialized endothelial cells, astrocytes, and parasites on a non-cellular basement membrane. The research states it diminishes the rate of access of many chemicals in plasma, thus preventing unencumbered access to the brain by circulating blood components. So essentially, it evolved to protect the brain from toxins.
Precisely. Think of the BBB as the brain's ultra-exclusive VIP club with the toughest bouncers imaginable. It's designed to keep almost everything out, protecting that delicate neural environment from circulating toxins, pathogens, and rapid fluctuations in blood chemistry. However, from the perspective of CNS therapeutics, this highly protective mechanism represents a substantial obstacle, a major hurdle for drug delivery to the site of action. Most pharmaceutical compounds simply cannot gain entry. The main exceptions are primarily lipophilic molecules or fat-soluble compounds. Because of their chemical properties, they can diffuse more freely across the BBB and accumulate in the brain. But for many other drug candidates, passage is determined by a complex combination of the solute's intrinsic properties, like its molecular weight, electrical charge, lipophilicity, and the presence or absence of highly specific energy-dependent transport systems embedded within the barrier cells.
So it's not just a passive filter. It actively manages what gets in and out. The cells forming the barrier also possess these essential transport systems that actively shuttle critical molecules like glucose and amino acids into the brain because they are critical for brain function. The research gives a great example. A transporter selected for large amino acids catalyzes the movement across the BBB and thus contributes to the therapeutic utility of L-DOPA in the treatment of Parkinson's disease. And for some compounds, like specific metabolites of neurotransmitters such as homophenolic acid and 5-hydroxy-indoleacetic acid, the acid transport system of the choroid plexus provides an important route for clearance from the brain, making sure waste products are efficiently removed.
Exactly right. The BBB is both the brain's indispensable bodyguard and, for drug developers, a major therapeutic headache. It's truly a double-edged sword when it comes to medicine. But understanding its intricate mechanisms, how it lets the good stuff in and keeps the bad stuff out, and how it actively clears metabolites is absolutely key. Key to overcoming those therapeutic challenges and designing drugs that can actually reach their targets within the CNS. 3. The electrical spark. Neuronal excitability, and ion channels.
Okay, now that we understand the brain's cellular players and its gatekeepers, let's talk about the action itself, the electrical spark that ignites neural communication. The research explains that neurons, these primary signaling cells, release neurotransmitters in response to a rapid rise and fall in membrane potential known as an action potential. And this all happens because voltage-dependent ion channels within the plasma membrane open when the membrane potential increases to a threshold value. This exquisitely controls the electrical excitability of neurons.
Action potentials are, quite literally, the neuron's fundamental language. They're the rapid electrical pulses that allow neurons to receive, encode, and transmit information across vast networks. And the core of this language lies in the incredibly precise symphony of ions. We now have a detailed understanding of how 3 major positively charged ions, sodium Na+, potassium K+, and calcium Ca2+, as well as negatively charged chloride, Cl anions, are meticulously regulated via their flow through highly selective ion channels embedded in the neuron's membrane.
So how do neurons maintain this delicate electrical equilibrium? I understand there's an active transmembrane countertransport of Na+ and K+ money and intracellular sequestration and mobilization of Ca2+ D2, which all work tirelessly to maintain those specific steady-state ion concentrations essential for electrical excitability.
Precisely. Think of it like a finely tuned battery that needs constant recharging. Voltage-sensitive ion channels are the gates that permit the swift flow of sodium and potassium ions. They mediate the rapid depolarization, making the inside more positive, and subsequent repolarization, making it negative again during an action potential. Then, powerful homeostatic mechanisms like the Na plus K plus ATPase pump, the Na plus CO2 plus exchanger, and Ca2 plus ATPases work tirelessly behind the scenes. They restore those precise transmembrane ionic gradients, ensuring the neuron is ready to fire again almost immediately. For instance, sodium is kept at a much higher concentration outside the cell, so when its channels open, it rushes in, causing rapid depolarization and initiating the action potential. Conversely, potassium is highly concentrated inside, so its outflow causes repolarization, getting the cell ready for the next signal. And calcium, while present at very low intracellular concentrations normally, plays a critical role. Even tiny changes in its intracellular level, say from 100 nanomolar up to 1 micromolar, affect multiple processes. And these changes are absolutely vital for triggering the release of neurotransmitters at the synapse. This whole intricate process of ion movement generates the electrical spark that's the very basis of neuronal communication.
And then there's chloride, the principal anion involved in neuronal excitability. The research shows chloride channels are essential for maintaining the neuron's resting potential and are responsible for the inhibitory postsynaptic potentials that actually dampen or break neuronal excitability.
That's spot on. In most mature neurons, the chloride concentration is kept much lower inside the cell than outside. So when chloride channels open, negatively charged chloride ions flow inward, making the inside of the neuron more negative. This is called hyperpolarization. This makes it harder for the neuron to reach its firing threshold. So consequently, if these chloride channels are inactivated, it leads to hyperexcitability because that inhibitory brake is effectively removed. There are several families of both voltage-gated and ligand-gated chloride channels. Crucially, the ligand-gated chloride channels are directly linked to key inhibitory neurotransmitters like gamma-aminobutyric acid, GABA, and glycine, which we'll definitely discuss in more detail shortly.
And it's not just the channels themselves. A class of secondary active transporters, the cation chloride cotransporters, plays an essential role in establishing the electrochemical chloride gradient needed for that hyperpolarizing inhibition mediated by GABA and glycine receptors. And this is truly astonishing During brain development, shifts in the expression of these cotransporter isoforms can actually reverse the chloride gradient, meaning that activating a ligand-gated chloride channel can become excitatory rather than inhibitory. That's a huge fundamental developmental shift in the brain's wiring.
It absolutely highlights the incredible plasticity and precision required for brain development, and the subtle yet profound changes that can occur. The CLC family of chloride channels also includes plasma membrane channels affecting chloride flux and membrane potential, as well as chloride hydrogen antiporters that influence the pH of intracellular vesicles, which is crucial for maintaining the internal environment of cellular compartments. And then there are the CFTR channels, or cystic fibrosis transmembrane conductance regulated channels. These are gated by ATP and activated by pKa, mediating the conductance of chloride and bicarbonate ions. Overall, these diverse chloride channels are responsible for a variety of important neurophysiological roles: regulation of membrane potential, maintenance of cellular volume, regulation of pH. It's a complex, finely tuned symphony of ions and their gates.
Let's move to cyclic nucleotide-gated, or CNG channels. These are described as non-selective cation channels that regulate ion flux in neurons and are activated by the binding of cyclic nucleotides like CAMP or cGMP. The research points to their primary function being quite specific, doesn't it?
Yes, their primary function really involves sensory transduction, especially in the retina, where they're key for vision, and in olfactory neurons, crucial for our sense of smell. Because they're non-selective and allow various alkali ions to flow, their activation can result in either depolarization or hyperpolarization. It depends on the specific ionic gradients and the cellular context. Structurally, they're composed of 4 subunits assembled around a central pore, typically classified into alpha and beta subunits.
And closely related are the hyperpolarization-activated cyclic nucleotide-gated, or HCN channels. These are another type of non-selective ligand-gated cation channel encoded by 4 different genes. I understand they're expressed in the heart as well as extensively in the brain, influencing how neurons respond to synaptic input.
That's correct. And HCN channels have been directly implicated in conditions like epilepsy and neuropathic pain. That underscores their critical role in regulating neuronal excitability. What's particularly unique about them is their activation mechanism. They open with hyperpolarization, meaning when the cell becomes more negative, and close with depolarization. The direct binding of cyclic AMP or cyclic GMP can further shift their activation curves to more hyperpolarized potentials, allowing for this kind of fine-tuned modulation. This intricate gating mechanism makes them essential in cardiac pacemaker cells, generating the heart's rhythm. And they're crucial in rhythmic and oscillatory activity within the CNS, as they are finely tuned to respond to specific activation thresholds, influencing processes like sleep and wakefulness.
Now, perhaps one of the most intriguing families of channels are the transient receptor potential, or TRP, channels. The name itself is a bit unfortunate, the research notes, because it doesn't immediately describe their incredibly diverse functions. But they're a large family, around 28 ion channels, that are non-selectively permeable to cations, including sodium, calcium, and magnesium. So, beyond their name, what do they actually do?
Their true function, despite the misnomer, is remarkably broad. They act as polymodal sensors, meaning they respond to multiple types of stimuli, allowing sequential entry into the cell. They're broadly grouped into 6 receptor subfamilies: TRPA, TRPC, TRPM, TRPML, TRPP, and TRPV, each with distinct activation profiles. These channels have incredibly diverse modes of activation and permeation. They play crucial roles across various aspects of sensory physiology, including thermosensation, our ability to detect temperature, osmosensation, and taste.
And this is where it gets really relatable and intriguing. Some TRP channels are also direct mediators of pain, acting as detectors of thermal and chemical stimuli that activate sensory neurons. For instance, the pungency we feel from common spices like garlic, chili powder, wasabi, that's due to their activation of certain TRP subtypes. Conversely, others respond to the familiar cool sensations from menthol, peppermint, and camphor.
Exactly. And beyond these fascinating sensory roles, the research indicates that mutations in TRP channels have been associated with both neurodegenerative diseases and cancer. That underscores their broad physiological importance beyond just sensation. The sheer diversity of their physiology has, very naturally, led to their investigation as promising drug targets. The capsaicin receptor, TRPV1, which famously gives chili peppers their heat, is the most thoroughly characterized TRP channel. Both agonists, molecules that activate it, and antagonists, molecules that block it, are being intensely investigated, particularly for the treatment of chronic pain. This raises an important point. How did we even begin to understand these ubiquitous channels? Well, the 2021 Nobel Prize in Physiology or Medicine was awarded to David Julius for his foundational work on TRP channels and to Artem Patapoutian for his work on mechanosensitive ion channels. It really highlights the profound importance of this research in decoding our senses and their link to disease. 4. The chemical dialogue. Neurotransmitters and synaptic transmission.
Okay, with the stage now set, understanding the cells and the electrical sparks, let's move into the truly profound chemical dialogue that underlies all brain function. A central principle in neuropsychopharmacology is that drugs aimed at improving the functional status of patients with neurological or psychiatric diseases. They typically act by either enhancing or blunting neurotransmission in the CNS. So specifically, where do these therapeutic agents exert their influence? What are the targets?
Right. Therapeutic targets are typically found at key molecular control points within that chemical communication pathway. This includes the ion channels, which we just discussed, that mediate the rapid changes in excitability induced by neurotransmitters. They also directly target the neurotransmitter receptors themselves. These are the proteins that physiologically recognize and respond to the chemical messages. And crucially, they target the transport proteins that efficiently clear-release neurotransmitters from the extracellular space, thus terminating their action. These are really the core molecular players that pharmacology seeks to modulate.
But before we dive into specific neurotransmitters, how do scientists even identify them in the first place? The research outlines this rigorous scientific checklist for identifying central neurotransmitters. They're described as endogenous chemicals in the brain that act to enable signaling across a chemical synapse. No, it's not enough to simply find a chemical in the brain, is it? You need to prove its function.
No, it's far more rigorous than that. Scientists had to develop this very stringent set of 5 criteria to confirm a substance's legitimate role as a true chemical messenger. First, the candidate transmitter must be present in the presynaptic terminals of the synapse and in the neurons from which those terminals arise. Second, it must be released from the presynaptic nerve along with nerve activity and in high enough quantity to have a discernible effect on the postsynaptic neuron. Third, the effects of experimentally applying the putative transmitter should precisely mimic the effects of stimulating the presynaptic pathway naturally. Fourth, if available, specific pharmacological agonists, which stimulate, and antagonists, which block, should reliably stimulate and block, respectively, the measured functions of the potential transmitter. And finally, there must be a clear mechanism present, either reuptake back into the cell or enzymatic degradation in the synaptic cleft, that effectively stops the actions of the transmitter. This ensures the signal is precisely controlled and doesn't just stay continuously active.
Okay, let's unpack this a bit.
Mm-hmm.
It's like finding a letter, but then you need to prove exactly who wrote it, who received it, and that it actually delivered a clear, specific message that was then, put away afterwards. The research also notes the inherent complexity. Many neurons contain multiple transmitter substances. They may act jointly or presynaptically. This often makes it difficult to mimic or fully antagonize a specific effect with just a single drug because the signaling is so nuanced, right?
It's a profound challenge, absolutely. Given that individual neurons can be incredibly sophisticated communicators, releasing multiple signals, this complexity in identifying the precise roles of signaling molecules has been partially overcome through the use of highly defined in vitro cell culture systems and advanced conditional transgenic animal models. These controlled environments allow scientists to isolate and study specific molecular interactions with unprecedented precision, and then try to extrapolate those findings back to the much more complex human CNS.
So, once a neurotransmitter is identified and its role confirmed, what are the actual fundamental stages of chemical communication, or synaptic transmission, that govern how neurons talk to each other?
Right, the entire process involves several highly specialized cellular functions, all happening in a tightly coordinated sequence. First, there's neurotransmitter synthesis. Small molecule neurotransmitters like dopamine or serotonin are typically synthesized rapidly right there in the nerve terminals. Larger molecules like peptides are synthesized in the neuron's cell body, the perikarya, and then transported down the axon to the terminals. Second, neurotransmitter storage. Once synthesized, these messengers are packaged into tiny, specialized containers called synaptic vesicles, often together with various proteins and ATP, ready for release. Third, and arguably the most critical step, is neurotransmitter release. This happens via a process called exocytosis. When the presynaptic neuron is sufficiently depolarized, it triggers a complex sequence. The synaptic vesicles dock at the plasma membrane, fuse with it, and then comes the calcium-dependent release of the vesicle contents, the neurotransmitters, into the synaptic cleft, that tiny space between neurons. Fourth is neurotransmitter recognition. Once released, the neurotransmitters rapidly diffuse across that cleft and bind selectively to specific receptor proteins located on the postsynaptic cell. This initiates intracellular signaling events within that receiving neuron. And finally, termination of action. A variety of crucial mechanisms quickly terminate the signal to ensure precise communication. This includes simple diffusion of the transmitter away from the synapse, enzymatic inactivation, where specific enzymes break down the transmitter, like with acetylcholine and peptides, and highly efficient reuptake of the transmitter back into the presynaptic neuron and/or neighboring glial cells by specialized transport proteins.
That's an incredibly detailed cascade. Now let's distinguish between two main speeds of communication in the brain: fast and slow neurotransmission. Fast neurotransmission is described as a very rapid, happening in milliseconds, because the effects are direct. These neurotransmitters bind directly to ligand-gated ion channels on the postsynaptic membrane to rapidly open the channel. What's the immediate result of that rapid opening, and what kind of receptors are we talking about here?
Right. The rapid opening of these channels directly changes the permeability of the postsynaptic membrane to specific ions. This leads to either depolarization or hyperpolarization. Depolarization pushes the neuron closer to its firing threshold, effectively transmitting and continuing the nerve impulse. Hyperpolarization, conversely, makes it less likely to fire, acting as an inhibitory brake, leading to diminished signaling. These ligand-gated ion channels, also known as iotropic receptors, are structurally composed of multiple protein subunits, typically 5 of them, pentamers. Each subunit usually has 4 transmembrane domains that come together to form the central pore. Prime examples include the nicotinic cholinergic receptors, activated by acetylcholine, the receptors for amino acids like GABA, glycine, glutamate, and aspartate, and the serotonin 5-HT3 receptor. The nicotinic H receptor is a particularly good example of how even slight variations in subunit composition can vary with anatomical location and profoundly affect the receptor's specific function and how drugs interact with it.
And then there's slow neurotransmission, operating on a much longer timescale, often seconds. Here, neurotransmitters do not bind to ion channels, but to receptors with a very different architecture that generates second messengers upon activation. Okay, great. These are the ubiquitous G protein-coupled receptors, or GPCRs. Could you describe their unique architectural marvel? They sound incredibly important.
They are. GPCRs are truly a masterpiece of biological engineering, and their structure is critical to their function. They all share this characteristic architecture of 7 transmembrane spanning domains. The protein chain weaves in and out of the cell membrane 7 times. They typically feature sites for N-linked glycosylation on their extracellular parts, which can influence their stability and interactions. Crucially, they possess multiple potential sites for phosphorylation, often by protein kinases like pKa and pKc, located on their intracellular loops and tail. This phosphorylation is vital because it can regulate GPCR-G protein coupling and provide docking sites for other proteins like arrestins and scaffolding proteins, which are important for modulating and turning off their signals. Some GPCRs are also palmitoylated, which can affect their flexibility and positioning within the membrane.
And there are a simply staggering number of them, aren't there? The research states 1,800 human GPCRs classified into 5 major families. What kind of broad physiological effects do these slower-acting but incredibly prevalent receptors mediate?
They mediate an extraordinarily broad spectrum of physiological effects, making them central to almost every bodily process imaginable. This includes things like the activation of potassium channels, the activation of phospholipase CYP3 calcium pathways, and the modulation of adenyl cyclase activity and all the downstream systems affected by cyclic AMP. These diverse effects are typically mediated through the activation of specific G proteins, which are heterotrimers composed of alpha, beta, and gamma subunits. The GTP-binding alpha subunits can then modulate numerous effector enzymes and ion channels, but the beta-gamma subunits are also active in mediating signaling, particularly in regulating specific ion channels. What's more, GPCRs aren't limited just to G protein signaling. They can also signal through other intermediary proteins, like the beta-arrestins. These play crucial roles in receptor regulation, desensitization, and even alternative signaling pathways.
The significance of GPCRs as drug targets really cannot be overstated. The research explicitly states that drugs targeting GPCRs represent a core of modern medicine and comprise over 1/3 of all pharmaceuticals. That's huge.
It is huge, and this raises an important question for us. Why is understanding these two fundamental speeds of communication, fast ionotropic and slow metabotropic, so critical? Not just for brain function, but for drug development too. Fast transmission, mediated by those ionotropic receptors, handles the rapid impulses, like immediate sensory processing or a quick reflex. But slow GPCR signaling, with its ability to generate second messengers and modulate cellular processes over seconds or even minutes, allows for much more subtle, long-lasting modulation of neuronal function. This impacts everything from our mood and emotional states to our capacity for memory and complex cognition. This sheer breadth of involvement offers an immense canvas for therapeutic intervention. It provides far more nuanced control points for addressing complex brain disorders than just relying on rapid on-off switches. 5. The messengers themselves. Central neurotransmitters in depth.
Okay, let's dive into the specifics of these fascinating chemical messengers now, starting with the amino acids. The research tells us they're found in high concentrations in the brain and have these rapid, readily reversible effects, and they're broadly categorized as either excitatory, sort of revving up brain activity, or inhibitory, applying the brakes.
That's correct. On the excitatory side, the main players are glutamate and aspartate, which are dicarboxylic amino acids. These are the brain's primary accelerators. On the inhibitory side, we find GABA, gamma-aminobutyric acid, glycine, valine, and taurine. These are monocarboxylic amino acids, the brain's brakes. The effects of glutamate and GABA are especially notable, just given their widespread and profound roles in nearly every brain function you can think of. And the development of selective pharmacological agonists and antagonists has been absolutely foundational. It allowed scientists to dissect and identify pharmacologically distinct amino acid receptor subtypes, which is a vital step for targeted drug development.
Let's dive deeper into glutamate and aspartate, the brain's chief accelerators then. Glutamate is described as the most abundant excitatory neurotransmitter and the principal fast excitatory neurotransmitter in virtually every region of the CNS. What are its main receptor types, and why is it so incredibly significant for brain function?
Right. Glutamate exerts its profound influence through both ligand-gated ion channels, the inotropic receptors, and metabotropic GPCRs, which are the MGOLRs. A critically important phenomenon involving glutamate is the induction of long-term potentiation, LTP, and its converse, long-term depression, LTD. These are the cellular mechanisms for strengthening and weakening synaptic connections, respectively, and they are widely hypothesized to be the fundamental processes underpinning learning, memory, and other adaptive processes in the brain.
And for the ionotropic glutamate receptors, they were historically classified into three types: NMDA, AMPA, and Ka receptors, named after their preferred synthetic ligands. How do NMDA receptors in particular function? They seem critical for learning.
NMDA receptors are truly unique. They're often called the brain's coincidence detectors. Think of them as gates that only open under specific, precise conditions. Structurally, they are heteromers, typically made up of multiple subunit combinations. The minimal functional receptor is usually a dimer of the GLUT2N1 subunit and at least one GLUT2N2 subunit. Functionally, they have a relatively high permeability to calcium ions, meaning they allow a significant influx of Ca2+ when they open. Crucially, they are blocked by magnesium in a voltage-dependent manner. This means at the neuron's normal resting potential, a magnesium ion physically sits in the channel, preventing ion flow even if glutamate is present. What's truly unique and defines them as coincidence detectors is that their activation requires the simultaneous binding of two different agonists. In addition to glutamate, glycine binding is absolutely necessary for the channel to open. So you need both the chemical signals and the right electrical state, depolarization, to dislodge that magnesium block. While they are involved in normal synaptic transmission, their activation is more closely associated with the induction of various forms of synaptic plasticity, the brain's ability to change and adapt rather than just mediating rapid point-to-point signaling.
Right. So aspartate is also a selective NMDA receptor agonist, and there are some well-known NMDA receptor ligands that act as open-channel blockers, like PCP and ketamine. There are also allosteric antagonists, like 5-7-dichloroquine acid at the glycine site, and ifenprodil, which selectively inhibits receptors containing GLUT2B subunits. Ketamine, that racemic mixture, has garnered significant clinical interest for its rapid antidepressant effects in drug-resistant depression. The S-enantiomer, esketamine, has even been introduced clinically, though there's ongoing discussion about its efficacy compared to the R-isomer or the mix. Its rapid effects are thought to be due, at least in part, to its NMDA receptor antagonism.
Exactly. And it's also vital to note that NMDA receptor activity is exquisitely sensitive to changes in pH and to modulation by a wide variety of endogenous agents. Things like zinc ions, certain neurosteroids, arachidonic acid, redox reagents, and polyamines like spermine can all influence their activity. This just shows how incredibly finely tuned their function is and how many natural compounds within the brain can potentially affect them.
Okay, then we have AMPA receptors. These exist predominantly as heterotramers, including various GLUAC subunits. They also interact with specialized transmembrane AMPA receptor regulatory proteins, or TARPs, and other scaffolding proteins that modulate their properties and trafficking to the synapse.
AMPA receptors are all about speed. They open and close incredibly rapidly within milliseconds. This makes them perfectly suited to mediate the vast majority of fast excitatory synaptic transmission in the brain, that quick, direct communication. Like NMDA receptors, they're also critically involved in synaptic plasticity. They can be selectively antagonized by compounds like MBQX and CNQX. These types of antagonists are currently being explored as potential neuroprotective drugs, for example, in treating stroke, where reducing excessive neuronal excitation could be vital to minimizing damage after blood flow is interrupted.
And finally, the Ka receptors, composed of distinct BlueKex subunits. The research highlights a key difference. Ka receptors require extracellular sodium and chloride for activation, which sets them apart from AMPA and NMDA receptors.
Functionally, Ka receptors differ in other important ways too. They don't predominantly reside within those main postsynaptic signaling complexes like AMPA receptors, mediating direct fast transmission. Instead, they seem to be strategically positioned to modulate neuronal excitability and synaptic transmission. They alter the likelihood that the postsynaptic cell will fire in response to subsequent stimulation. Presynaptic Ka receptors have also been implicated in modulating GABA release, further highlighting their more nuanced modulatory role rather than a purely direct excitatory one.
This brings us to a truly critical concept: glutamate-mediated excitotoxicity. This phenomenon is thought to underlie the neuronal damage that occurs when conditions like ischemia or hypoglycemia in the brain lead to a massive uncontrolled release and impaired reuptake of glutamate. The result is this excess stimulation of glutamate receptors and subsequent, often irreversible, cell death.
Yes, the cascade leading to neuronal death and excitotoxicity is profound and multi-layered. It's quite devastating. It appears to be triggered by excessive, sustained activation of either NMDA or AMPA receptors. This allows an overwhelming influx and mobilization of calcium into neurons. This calcium overload then promotes significant oxidative stress within the cell. This leads to an increase in harmful molecules like nitric oxide and peroxynitrite, a decrease in protective compounds like glutathione, and a surge in reactive oxygen species. This, in turn, results in lipid peroxidation, destabilization of lysosomes, severe damage to mitochondria, the cell's powerhouses, ultimately leading to cell death via a programmed process called apoptosis. The good news, therapeutically speaking, is that NMDA receptor antagonists have shown the ability to attenuate neuronal cell death induced by this overactivation. This makes glutamate receptors critically important targets for diverse therapeutic interventions, particularly in understanding and potentially treating chronic neurodegenerative diseases where excitotoxicity might play a role.
Beyond the ionotropic receptors, we have the metabotropic glutamate receptors, or MGLRs. The research describes these as the brain's fine-tuners. These are GPCRs with a distinctive structure, featuring a large N-terminal domain that contains the orthosteric binding site for glutamate. They function as obligate dimers, meaning they must pair up to be active.
That's right. Orthosteric agonists, including glutamate itself, and antagonists bind to that large N-terminal domain. While allosteric modulators, molecules that bind elsewhere to change the receptor shape and function, typically interact within the 7-transmembrane spanning domain. Once activated, G proteins and beta-arrestins engage the cytoplasmic surface of the mGLUR, allowing for diverse downstream signaling cascades. There are 8 unique mGLURs identified, organized into 3 subgroups based on sequence homology, pharmacology, and signaling pathways. Group I mGLURs, mGLUR1 and mGLUR5, coupled to GQ activation and generally increase neuronal excitability. Groups 2 and 3, mGLUR3, mGLUR4, mGLUR6, mGLUR7, and mGLUR8, coupled to GGO proteins and typically suppress excitability. They are distributed throughout various brain regions and play crucial roles in fine-tuning excitatory and inhibitory transmission through presynaptic, postsynaptic, and even glial mechanisms. They are also known to modulate the release and signaling of other neurotransmitters, like GABA, purines, dopamine, serotonin, and various neuropeptides. Beyond that, they play a known important role in synaptic plasticity, influencing learning and memory.
And this sounds like a very promising area for modern drug development. The widespread expression of MGLRs, their capacity to modulate presynaptic neurotransmission in this dynamic and activity-dependent way, and the increasing availability of subtype selective modulators, both negative allosteric modulators, lean names, that decrease activity, and positive allosteric modulators, PAMs, that increase it, make them highly attractive targets, especially for treating cognitive impairment related to several psychiatric and neurodegenerative diseases. What are some specific examples of their therapeutic potential?
There are indeed many exciting possibilities that highlight this precision. For example, MDLR5 is highly involved in anxiety disorders. Its activation increases neuronal excitability and NMDA receptor currents. Consistent with this, MDLR5 and AIMS molecules that reduce the receptor's activity have shown robust efficacy in animal models of anxiolytic activity. And the clinically validated anxiolytic agent phenobam actually acts as a selective MDLR5 NAM. Another promising area involves group 3 mGLRs, which include mGLR4, mGLR7, and mGLR8. These are expressed in both GABAergic and glutamatergic terminals. Agonists and PAMs for these receptors can inhibit the release of both glutamate and GABA in Parkinson's disease, thereby opposing glutamate hyperactivity. Specifically, MDR4 PAMs reduce motor symptoms in animal models of Parkinson's disease and may play a role in neuroprotection and enrichment of motor responses to low doses of L-DOPA. Similarly, inhibiting presynaptic glutamate release has been implicated as a therapeutic strategy for other conditions linked to excitation toxicity like Huntington's disease. And there's growing evidence that group 2 MDLRs may play a significant role in the cognitive decline observed in schizophrenia. What's truly fascinating here is how these subtle, finely tuned modulations offer such precise control. It allows for a level of therapeutic finesse that single-target sort of brute force drugs might not achieve. It's really a leap forward in understanding and potentially treating complex brain disorders.
Okay, let's switch gears now to the main inhibitory neurotransmitter in the CNS, gamma-aminobutyric acid, or GABA. Our brains absolutely need brakes as much as accelerators. How is GABA synthesized, and how is its action controlled?
Right. GABA synthesis is intricately linked to the Krebs cycle, that central metabolic pathway. It starts with iketoglutarate, which is transaminated to glutamate by the enzyme GABA transaminase, or GABA-T. Then GABA is subsequently formed directly from glutamate by the enzyme glutamic acid decarboxylase, or GAD. Interestingly, intraneuronal GABA is also inactivated by GABA-T, making it both a synthesis and degradative enzyme in what's known as the GABA shunt. This pathway serves to maintain optimal GABA levels. Once synthesized, GABA is stored in vesicles by a specific vesicular GABA transporter, VGAD. Its action in the synaptic cleft is primarily terminated by rapid reuptake via one of 4 different GABA transporters, GAD-cateates, which are present on both neurons and neighboring glial cells. This ensures precise control over its duration of action.
And GABA acts through 2 main receptor types, inotropic GABA receptors and metabotropic GABA receptors. The GABA receptors are extensively characterized as incredibly important drug targets. They're the site of action of many well-known neuroactive drugs, right? Like benzodiazepams, diazepam, for example, barbiturates, ethanol, anesthetic steroids, and volatile anesthetics.
Yes, these drugs are indeed widely utilized to treat a variety of neuropsychiatric disorders. Things like epilepsy, Huntington's disease, various addictions, and sleep disorders. Structurally, GABA receptors are pentamers, meaning they are composed of 5 protein subunits, each containing 4 transmembrane domains arranged around a central chloride-selective pore. The major functional forms typically contain alpha, beta, and gamma subunits, with a likely stoichiometry of 2 alpha, 2 beta, and 1 gamma. The diversity is astounding. The International Union of Basic and Clinical Pharmacology, IUPHAR, recognizes 19 unique subunits that can combine to form at least 11 native GABA receptors, each with distinct pharmacological properties. Crucially, the specific combination of alpha and gamma subunits can actually affect the efficacy of benzodiazepine binding and subsequent channel modulation, highlighting the precision required for drug design. Many of these clinically important drugs, like benzodiazepines and volatile anesthetics, act as positive allosteric modulators, PAMs, meaning they bind at a site distinct from the natural GABA binding site but enhance the receptor's function, making it more sensitive to GABA. The field has even seen amazing breakthroughs in understanding these interactions. High-resolution cryo-electron microscopy structures of the 1-per-tau-3 GABA receptor bound to the agonist GABA and the classical benzodiazepines, aprazolam and diazepam, have been solved. This gives incredible atomic-level insights, and a very recent development is brexanolone, a neuroactive steroid, and GABA-PAM, which has been FDA approved specifically for treating postpartum depression, showcasing the ongoing clinical relevance of targeting these receptors.
And here's where it gets really interesting with GABA. It's all about control. Think of it like the brain's dimmer switch, right? And different drugs can either gently dial down the activity, like a benzodiazepine reducing anxiety, or completely hit the off switch, like some anesthetics. The precision is incredible. And then we have GABA receptors, the slower-acting inhibitors.
GABA receptors are metabotropic GPCRs. And unlike many GPCRs, they function as obligate heterodimers, meaning they must consist of two distinct subunits, named GABA1 and GABA2, working together to be functional. They are widely distributed throughout the CNS and play critical roles in regulating both pre- and postsynaptic activity. These receptors primarily interact with G proteins, which leads to a cascade of inhibitory effects, inhibiting adenyl cyclase, activating certain potassium channels, and reducing calcium conductance. They can also, in some cases, interact with GQ proteins to enhance phospholipase C activity. Presynaptic GABA receptors often function as autoreceptors. They're located on the GABA-releasing neuron itself and act to inhibit its own GABA release, a feedback mechanism. They may also play a similar role on neurons releasing other transmitters, acting as heteroreceptors in that case. Clinically, agonists like baclofen, a well-known skeletal muscle relaxant, and gamma-hydroxybutyric acid, GHB, used for narcolepsy and also known as a recreational intoxicant, demonstrate their diverse therapeutic and physiological relevance.
Next up, glycine. This amino acid is an inhibitory neurotransmitter, particularly prominent in the spinal cord and brainstem. It's synthesized primarily from serine by serine hydroxymethyltransferase and, much like GABA, enters synaptic vesicles via VJAT, the same transporter.
That's correct, and glycine's action in the synaptic cleft is efficiently terminated by reuptake through specific transporters, GLYT1 and GLYT2, located on presynaptic nerve terminals and glial cells. These transporters are increasingly seen as attractive therapeutic targets for modulating glycine levels, especially given glycine's fascinating dual role. It also acts as a required coagonist at NMDA receptors alongside glutamate for their activation. In addition to this modulatory role at NMDA receptors, there are specific ionotropic glycine receptors. These are pentamers of subunits containing 4 transmembrane domains, and they function as hyperpolarizing chloride channels, primarily in the brainstem and spinal cord. These receptors comprise 4 types of alpha subunits and 1 beta subunit, forming various functional receptor subtypes. Specific pharmacological agents include taurine and alanine, which act as agonists, while strychnine, a potent neurotoxin, is a highly selective antagonist, demonstrating the precision with which these receptors can be targeted.
Now let's talk about acetylcholine. AC? Famously the first neurotransmitter discovered. It occurs throughout the nervous system and plays really broad roles.
AC is absolutely fundamental to communication across vast parts of the nervous system. It plays a primary role in the autonomic nervous system, mediating both ganglionic transmission and parasympathetic innervation of organs. It's also the neurotransmitter at the somatic motor nerves that innervate skeletal muscle, enabling all our voluntary movement. In the CNS, it's primarily found in interneurons and in specific projection pathways originating from brainstem and basal forebrain nuclei. Acet is synthesized by choline acetyltransferase and stored in vesicles. After release and receptor activation, it's rapidly and efficiently degraded by acetylcholinesterase in the synaptic cleft. This ensures precise, short-lived signaling. Clinically, the profound degeneration of these cholinergic pathways is a key hallmark pathology of Alzheimer's disease, making this system a hugely significant area of therapeutic research.
And HE exerts its effects through 2 major classes of receptors. First, the nicotinic HE receptors. These are ligand-gated ion channels found not just at the neuromuscular junction, but also widely in autonomic ganglia, the adrenal gland, and critically throughout the CNS. Their activation by HE results in a rapid increase in sodium influx, leading to depolarization and also calcium influx.
Nicotinic receptors are pentamers, meaning they are composed of 5 protein subunits, which can be various combinations of 5 out of 17 known subunits. In the CNS, they are typically assembled as combinations of alpha-210 and beta-24 subunits, or sometimes even as a homomeric alpha-7 receptor, where all 5 subunits are identical alpha-7 proteins. The precise subunit composition profoundly influences their biophysical and pharmacological properties. It dictates their specific function and drug sensitivity. These receptors have incredibly high therapeutic value, not only for smoking cessation, as they are the primary receptors targeted by nicotine, but also for potentially modulating other neurological pathologies where cholinergic signaling is implicated.
And then the muscarinic HE receptors, which are all GPCRs.
Yes, there are 5 distinct subtypes of muscarinic receptors, M1 through M5, and all are extensively expressed in the brain. They exhibit different G protein coupling, which leads to different downstream effects. M1, M3, and M5 primarily couple to GQ proteins, leading to the activation of phospholipase C and related signaling pathways. In contrast, M2 and M4 receptors couple to GGO proteins, primarily inhibiting adenylate cyclase activity. This diversity in subtype and G protein coupling allows muscarinic receptors to mediate an incredibly wide range of physiological effects, from regulating heart rate to modulating cognition and memory. This makes them important targets for a variety of neurological conditions.
Moving on to the monoamines. These are a diverse group of neurotransmitters characterized by an amino group connected to an aromatic ring by a two-carbon chain, and they are derived from aromatic amino acids. The research indicates they regulate neurotransmission that underlies cognitive processes, including emotion.
Indeed. And drugs that affect monoamine receptors and their signaling pathways are among the most widely used pharmacotherapies globally. They target a broad spectrum of conditions from depression, schizophrenia, and anxiety, all the way to debilitating movement disorders like Parkinson's disease. This group includes dopamine, norepinephrine, epinephrine, histamine, serotonin, and the trace amines. Each monoamine system is anatomically distinct, originating from specific clusters of neurons, mostly in the brainstem, and projecting broadly throughout the brain. They serve separate, yet often interconnected, functional roles within their fields of innervation. This anatomical and functional specificity contributes immensely to the complexity and precision of brain function, and consequently to the challenges and opportunities in drug development targeting these systems.
Let's start with dopamine, DA, which is the predominant catecholamine in the CNS. What are its major pathways and what fundamental roles does it play in the brain?
There are three major, functionally distinct dopamine-containing pathways in the CNS that mediate its widespread effects. The nigrostriatal pathway is crucial for motor control, and its degeneration is the hallmark pathology of Parkinson's disease. The mesocortical mesolimbic pathway is doubly involved in motivation reward, which is why most drugs of abuse potently increase dopamine signaling and emotional regulation. And the tuberoinfundibular pathway plays a key role in regulating the release of various hormones, particularly prolactin, from the pituitary gland. These diverse physiological effects are mediated by 5 distinct GPCRs grouped into 2 subfamilies. There are the D1-like receptors, D1 and D5, which primarily stimulate adenylate cyclase activity via coupling to Gs or Gulf proteins, generally leading to excitatory effects, and the D2-like receptors, D2, D3, and D4, which couple to GGO proteins to inhibit adenylate cyclase activity and modulate various voltage-gated ion channels, generally leading to inhibitory or modulatory effects. Dopamine pathways and their receptors have been profoundly implicated in the pathophysiology of both schizophrenia and Parkinson's disease, and critically also in the often challenging side effects that follow the pharmacotherapy of these disorders.
Then we have norepinephrine, NE. Both alpha and beta adrenergic receptor subtypes are present in the CNS and all are GPCRs.
Norepinephrine mediates its effects through a variety of adrenergic receptor subtypes. Beta adrenergic receptors, like RE223, primarily couple to G's proteins, activating adenylate cyclase and generally leading to excitatory or facilitatory effects, influencing things like arousal and attention. Alpha-1 adrenergic receptors couple to GQ proteins, stimulating the phospholipase C, PLC, IP3-diacylglycerol calcium PKC pathway, and are predominantly associated with neurons. When activated, they typically produce depolarizing responses due to decreases in potassium conductance. Alpha-2 adrenergic receptors are unique. They're found on glial cells, vascular elements, and importantly, on neurons. They are prominently located on noradrenergic neurons themselves, where they couple to G proteins, inhibit adenylate cyclase, and mediate a hyperpolarizing response by enhancing an inwardly rectifying potassium channel. Alpha-2 adrenergic receptors also function crucially as inhibitory autoreceptors located presynaptically. They detect their own neurotransmitter, Ne, and upon activation diminish the further release of Ne. The antihypertensive effects of the drug clonidine, for example, are thought to result in part from the stimulation of such autoreceptors in the brainstem, reducing sympathetic outflow from the CNS. Ne is widely distributed in the brain, with relatively large amounts in the hypothalamus and limbic system, like the amygdala and dentate gyrus. Noradrenergic neurons originating from the locus coeruleus broadly innervate most brain regions, highlighting its widespread modulatory influence on alertness, attention, and stress responses.
And epinephrine, EPI, is generally considered a minor catecholamine neurotransmitter in the brain compared to dopamine and norepinephrine, correct?
That's accurate. Most epinephrine found in the brain is actually contained within vascular elements, consistent with its primary role as a circulating hormone produced by the adrenal gland. Neurons containing epinephrine are quite rare and seem to be found in restricted connections, primarily within the medullary reticular formation and some pontine and diencephalic nuclei. Consequently, their precise physiological properties and roles specifically within the CNS have not yet been unambiguously identified. They remain a less understood monoamine in the context of brain-specific neurotransmission.
Now, histamine. Most of us know it for its role in allergies and inflammation, but it's also a significant monoamine neurotransmitter in the CNS. Where are these neurons found, and what functions does the histaminergic system affect in the brain?
Indeed, histamine's role extends far beyond allergies. Histaminergic neurons are located primarily in a specific area, the tuberomammillary nucleus of the ventral posterior hypothalamus, and they give rise to long ascending and descending tracts that broadly innervate the entire brain, much like the other major monoamergic systems. The histaminergic system is thought to profoundly affect crucial CNS functions like arousal, body temperature, and vascular dynamics. While a dedicated specific reuptake transporter for histamine hasn't been identified after its release, it is taken up by facilitative organic cation transporters, OCT2, OCT3, and the plasma membrane monoamine transporter, PMT. Histamine is then primarily metabolized by the enzyme histamine N methyltransferase. It signals through 4 distinct GPCR subtypes, H1 through H4, which regulate either adenylate cyclase or phospholipase C, leading to diverse cellular responses.
The H1 receptors are widely distributed in the brain, with high densities in regions linked to neuroendocrine functions, behavioral control, and nutritional state control.
That's right. Activation of H1 receptors typically excites neurons in most brain regions. Genetic knockout of the H1 receptor in animal models leads to significant behavioral state abnormalities, consistent with its major role in the cortical control of the sleep-wake cycle. This precisely explains the well-known sedative actions of first-generation H1 receptor blockers, those common antihistamines used for allergies because they cross the blood-brain barrier. Consequently, the pharmaceutical industry has focused heavily on developing second-generation H1 antagonists with low CNS penetration to reduce the sedation, allowing effective allergy treatment without making people drowsy. H2 receptors, while primarily known for their role outside the brain in gastric acid secretion and smooth muscle relaxation, and historically a mainstay for treating dyspepsia and acid reflux, are also highly expressed in the brain. Here they seem to regulate neuronal physiology and plasticity. Mice lacking H2 receptors show cognitive deficits and impaired hippocampal long-term potentiation, along with abnormalities in nociception or pain perception. While systemic H2 inhibition has demonstrated some anti-nociceptive effects, no H2 receptor antagonist has yet been approved specifically for pain management in humans.
And then H3 receptors, also present in the CNS. The research states they act as autoreceptors on histaminergic neurons to inhibit histamine synthesis and release.
That's a crucial negative feedback role. They also inhibit adenyl cyclase and modulate N-type voltage-gated calcium channels. Crucially, H3 receptors are not confined just to histaminergic neurons. They also act as heteroreceptors to regulate the release of other neurotransmitters like serotonin, acetylcholine, norepinephrine, and dopamine. As a consequence of this broad modulatory ability, the H3 receptor has become a highly attractive therapeutic target, potentially for treating various conditions such as obesity, movement disorders, schizophrenia, ADHD, and disorders of wakefulness. A wide array of compounds targeting H3 have been developed, and one, pitolisant, which functions as an inverse agonist at the H3 receptor, meaning it reduces the receptor's baseline activity, is FDA approved for treating narcolepsy, a sleep disorder. Finally, H4 receptors are mainly expressed on hematopoietic cells, blood cells, with limited expression on microglia within the CNS. Their primary known roles relate more to allergy, asthma, and possibly itching antipyretic properties.
Serotonin, 5-HT, is next. This one is widely known, especially for its profound role in mood, making it a critical target for many psychiatric medications.
Yes, absolutely. There are incredibly diverse pathways mediating serotonin signaling, and they play fundamental roles in modulating mood, depression, anxiety, and phobia. They also influence gastrointestinal motility and many other bodily functions. These widespread effects are mediated by a remarkably complex family of receptors. There are 13 distinct GPCR subtypes identified, plus one ligand-gated ion channel, the 5-HT3 receptor. Each of these subtypes exhibits characteristic ligand-binding profiles, couples to different intracellular signaling systems, and displays subtype-specific distribution within the CNS and periphery. This immense complexity and diversity within the serotonin system allows for highly precise and nuanced therapeutic targeting. Different drugs can preferentially activate or block specific receptor subtypes to achieve desired clinical outcomes in treating mood disorders, anxiety, and other conditions.
And then the emerging messengers, trace amines. The research says they've only recently been appreciated as neurotransmitters. They're detected at trace levels with very short half-lives due to rapid metabolism by monoamine oxidase. So what are these elusive messengers?
That's right. These are a fascinating, relatively new class of signaling molecules. Some trace amines indeed act as neuromodulators or even neurotransmitters interacting with specific receptors. They are structurally related to the catecholamines we discussed. Examples include phenethylamines like phenethylamine itself and tyramine, as well as tryptamine, octopamine, and synephrine. They are thought to exert their effects primarily through a family of GPCRs, originally called trace amine receptors, now more accurately known as trace amine-associated receptors, or TARs. The first such receptor was only identified back in 2001. So far, 6 human genes have been identified in this family, including TAR1. Interestingly, many TARs are prominently expressed in the olfactory epithelium, suggesting they might serve as olfactory receptors for volatile amines, contributing to our sense of smell. TAR1, also known as T1, the only one currently recognized by IUPAR for its clear role in the brain, is a GPCR that couples to Gs and G13 proteins. It has the highest affinity for endogenous trace amines like tyramine, methylphenephrine, and octopamine. Uniquely, in neurons, TAR1 seems to be predominantly intracellular. It can activate downstream signaling pathways from an intracellular membrane compartment, not just the cell surface like most GPCRs.
So it's not just on the cell surface. And the research indicates it modulates monoaminergic activity in the CNS and is activated by amphetamine and related psychostimulants, as well as endogenous compounds called thyronamines.
Exactly. This functional profile makes TAR1 a truly fascinating therapeutic target. Agonists for TAR1 have shown very promising and favorable effects in animal models of addiction and schizophrenia. This suggests a potentially groundbreaking new avenue for treatment. However, human therapeutics specifically targeting TAR1 have yet to be fully developed and brought to clinical fruition. It highlights an exciting frontier in CNS pharmacology.
Moving beyond the more conventional small molecule neurotransmitters, we have peptides. The research notes they typically behave as modulators of neurotransmission rather than direct agents of excitation or inhibition. What diverse functions do they have, and how do they differ in their biosynthesis and signaling compared to, say, glutamate or acetylcholine?
Peptides are an incredibly diverse class of signaling molecules, and their modulatory roles are indeed profound. They are involved in a wide array of fundamental brain functions, ranging from analgesia, pain relief, to social behaviors, complex learning, and memory formation. Unlike small molecule neurotransmitters, peptide biosynthesis is much more complex. It requires the transcription of DNA into mRNA and then the translation of mRNA into protein. This primarily occurs in the neuron's cell body, or pericardia. The synthesized peptide is then transported down the axon to the nerve terminals. What's more, a single gene can often give rise to multiple distinct neuropeptides through sophisticated transcriptional and post-translational modifications. For example, the propiomelanocortin POMC gene, just one genetic blueprint, gives rise to several distinct peptides, including ACTH, various melanocyte-stimulating hormones, MSH, and the opioid peptide endorphin. Even alternative splicing of RNA transcripts in different tissues can produce distinct mRNA species encoding different peptides, like calcitonin and calcitonin gene-related peptide, CGRP, all from the same initial gene. It's incredibly efficient.
And I understand most are thought to act in concert with coexisting neurotransmitters, often packaged into vesicles and released alongside other neurotransmitters to modulate their actions, so they add another layer of complexity.
Precisely. This co-release allows for incredibly sophisticated and nuanced signaling. While classical small molecule neurotransmitters generally signal by rapidly depolarizing or hyperpolarizing neurons to mediate quick communication, neuropeptides have far more diverse mechanisms of action. They can, for instance, affect gene expression within the target cell, leading to much longer-lasting effects on neuronal function. Their action is also terminated differently. They are not terminated by rapid reuptake back into the presynaptic cell like monoamines or amino acids. Instead, they are enzymatically inactivated by extracellular peptidases, which are enzymes that break them down in synaptic cleft or extracellular space. This slower inactivation process results in effects that are typically prolonged in nature compared to the fleeting actions of classical neurotransmitters.
Most neuropeptide receptors are GPCRs, with extracellular domains playing a primary role in peptide ligand binding. But the research also states that peptides themselves are typically inefficient as drugs, especially at CNS targets behind the formidable blood-brain barrier due to delivery difficulties. How is drug development tackling this challenge to unlock their therapeutic potential?
This has indeed been a major hurdle for decades. Given the difficulty of peptides crossing the BBB and their susceptibility to enzymatic degradation in the body, major efforts have historically focused on developing small molecule drugs that are orally bioavailable and can effectively cross the barrier, acting as either agonists or antagonists at neuropeptide receptors. Through a sophisticated combination of structural biology, advanced chemistry, high-throughput screening, HTS, and rational drug design, we now actually have small molecule ligands for many neuropeptide receptors. This has been a significant success story. While natural products haven't traditionally been a rich source of drugs for peptidergic transmission, a notable exception is the plant alkaloid morphine, which acts selectively at opioid receptor subtypes, demonstrating that nature can indeed provide blueprints. And now there's a truly revolutionary new avenue emerging: antibody-based therapeutics. The research highlights calcitonin gene-related peptide, CGRP, a neuropeptide that plays a key role in the development of migraine headaches. In a significant breakthrough, small molecule CGRP receptor antagonists, antibodies developed against CGRP itself to neutralize it, and antibodies developed against the CGRP receptor have all been approved as effective treatments for the prevention of migraine. This provides a completely new and incredibly promising approach for targeting peptidergic systems. It kind of sidesteps the BBB issue by using larger antibody molecules that don't need to enter the brain to exert their effects, often targeting peripheral components of the migraine pathway. If we connect this to the bigger picture, the shift from seeing peptides as mere signals to understanding them as sophisticated modulators with prolonged effects, combined with the advent of these antibody-based treatments, has opened up entirely new therapeutic strategies, especially for conditions like migraine.
Now let's explore purines. The research tells us that adenosine, ATP, UDP, and UTP molecules, primarily known for their roles in energy currency within cells, also have critical roles as extracellular signaling molecules. ATP, for example, is actually a component of many neurotransmitter storage vesicles and is released along with other transmitters. That's surprising.
It is quite surprising, but yes. Extracellular adenosine, a purine nucleoside, can also result from the cellular release and subsequent metabolism of ATP. ATP is rapidly hydrolyzed extracellularly by enzymes called ectonucleotidases. These extracellular nucleotides and adenosine can then act on a diverse family of purinergic receptors. And their signaling has been implicated in a variety of vital functions, including memory and learning, locomotor behavior, and feeding regulation.
And there are three major classes of purinergic receptors: adenosine receptors, P1, P2I, and P2X. Let's start with the adenosine receptors.
Adenosine receptors are all GPCRs. There are 4 distinct subtypes: A1, A2A, A2B, and A3, all of which are activated endogenously by adenosine. Their G protein coupling differs, leading to varied effects. A1 and A3 receptors primarily couple to G proteins, generally leading to inhibitory effects. In contrast, A2A and A2B receptors couple to G proteins, which typically leads to excitatory or facilitatory effects. Functionally, activation of A1 receptors can inhibit adenylyl cyclase, activate certain potassium currents, and in some instances activate phospholipase C. Stimulation of A2 receptors, on the other hand, activates adenylyl cyclase. In the CNS, both A1 and A2A receptors are crucially involved in regulating the release of other major neurotransmitters such as glutamate and dopamine. This makes the A2A receptor a significant potential therapeutic target for disorders such as Parkinson's disease. In fact, the A2A receptor antagonist, isradipine, was recently approved as an adjunctive therapy with L-DOP for the treatment of Parkinson's disease, highlighting its clear clinical utility.
Then there are P2Y receptors.
P2I receptors are also a family of GPCRs, but they are activated by a broader range of purine and pyrimidine nucleotides, including ATP, ADP, UTP, UDP, and even UDP glucose. There are 8 known subtypes that couple to various G proteins, leading to diverse intracellular signaling. For example, the P2Y14 receptor is expressed in the CNS, is stimulated by UDP glucose, and may play a role in neuroimmune functions. The P2Y12 receptor, while widely expressed, is particularly important clinically because its antagonism in platelets effectively inhibits platelet aggregation. This makes it a key target in cardiovascular medicine for preventing blood clots.
And finally, P2X receptors. These are described as ATP-sensitive ligand-gated cation channels expressed throughout the CNS on presynaptic and postsynaptic nerve terminals and on glial cells. So these are channels, not GPCRs.
Correct. P2X receptors are fundamentally different from the other purinergic receptors because they are ion channels, mediating rapid responses to ATP. They are notably found on nociceptive sensory neurons, which are crucial for pain sensation. Here, they primarily gate the flow of sodium, potassium, and calcium ions and are strongly implicated in mediating sensory transduction, basically converting stimuli into electrical signals. There are 7 subtypes, P2X1 through P2X7, with varying sensitivities to ATP. Structurally, P2X receptors have a trimeric topology, meaning they exist as either homopolymers, 3 identical subunits, or heteropolymers, 3 different subunits coming together. A major breakthrough in understanding these receptors was the first crystal structure confirming this trimeric arrangement back in 2009. Since then, more than 20 crystal structures have been described, dramatically deepening our understanding of their function and how drugs might interact. The study of selective compounds targeting specific P2X subtypes suggests that modulating these receptors may be profoundly useful in the therapy of neurological disorders such as neuropathic and inflammatory pain, epilepsy, memory disorders, and even alcohol use disorders.
And a P2X drug has actually made it to clinical fruition. Givapixant, a P2X3 receptor antagonist, was approved in Japan in 2022 for refractory chronic cough.
That's a fantastic example of directly translating this fundamental knowledge into a clinical therapy. It really shows the power of this research. The mechanism here is quite elegant. The release of ATP from irritated bronchial cells, for instance during chronic inflammation causing a cough, can stimulate P2X3 receptors on sensory nerve fibers in the airway. This initiates an action potential that directly leads to the coughing reflex. Givapixant works by inhibiting ATP binding to these P2X3 receptors, thereby reducing the stimulation that causes persistent coughing. And as a truly cool, respectful fact, it's named in part after the late Jeff Bernstock, a real pioneer of purinergic signaling, who first proposed purines as neurotransmitters way back in the 1970s. It's a wonderful nod to that scientific journey of discovery.
Let's discuss cannabinoids. These have certainly captured public attention, especially for their psychoactive effects. The identification of THC, delta-9 tetrahydrocannabinol, as the primary psychoactive substance in marijuana back in the 1960s ultimately led to the discovery of 2 cannabinoid receptors, CB1 and CB2, and eventually the endogenous compounds that naturally interact with them in our bodies.
Right. These 2 receptor subtypes, CB1 and CB2, are both GPCRs that primarily couple to Jago proteins. This coupling leads to a range of inhibitory effects. They typically inhibit adenyl cyclase, and in some cell types, they inhibit voltage-gated calcium channels or stimulate certain potassium channels, generally acting to dampen neuronal activity. Although they share relatively low overall amino acid homology, meaning their protein sequences aren't super similar, they are found in strikingly different locations. CB1 receptors are found in incredibly high levels throughout the brain, particularly in areas involved in cognition, memory, mood, appetite, and motor control. They mediate most of the psychoactive effects associated with cannabis use. In contrast, CB2 receptors are much more prominent in immune cells throughout the body. In the CNS, they are less abundant and found primarily on microglia, the brain's immune cells, suggesting a role in neuroinflammation and immune responses within the brain. The comprehensive discovery of the endogenous cannabinoid system, which includes these cannabinoid receptors, the body's own endogenous cannabinoids or endocannabinoids, and the enzymes that synthesize and degrade them, along with extensive clinical data from marijuana use, has profoundly expanded our understanding of this unique signaling system's complex physiology and therapeutic potential.
And the endocannabinoids themselves are lipid molecules like anandamide and 2-arachidonyl glycerol, 2-AG, plus other putative compounds that might also play roles.
Their actions are primarily terminated by a 2-step process. First, by uptake into cells, followed by hydrolysis or enzymatic breakdown within the cell. 2 key enzymes involved are FAAH, fatty acetylcholine hydrolase, which mainly breaks down anandamide, and MGL, monoacylglycerol lipase, which primarily breaks down 2-AG. While some earlier studies suggested a specific transport system might exist for endocannabinoids, no definitive molecular entity has been identified as a dedicated transporter yet. This is still an area of active research. However, this insight into their degradation directly leads to a promising therapeutic strategy. Drugs that inhibit the transport or degradation of endocannabinoids would effectively prolong their physiological actions. This would enhance the body's natural cannabinoid signaling, which could be beneficial for conditions ranging from chronic pain and inflammation to anxiety and potentially other disorders.
Beyond these, the research describes other lipid mediators that behave as unconventional neurotransmitters or modulators. For example, arachidonic acid, typically stored in cell membranes, can be liberated during phospholipid hydrolysis by enzymes like phospholipase A2.
Exactly. And once liberated from the cell membrane, arachidonic acid is not just a passive molecule. It can be converted into a whole range of highly reactive modulators by three major enzymatic pathways. These include cyclooxygenases, COX enzymes, which produce prostaglandins and thromboxane, involved in inflammation and blood clotting. Lipoxygenases, LOX enzymes, leading to leukotrienes, also involved in inflammation and asthma. And cytochrome P450s, CYP450 enzymes, which, while inducible and generally found at low levels in the brain, can also metabolize arachidonic acid into various signaling molecules. These various arachidonic acid metabolites have been compellingly implicated as diffusible modulators in the CNS. They might be involved in the formation of long-term potentiation and other forms of neuronal plasticity, suggesting a role in long-lasting changes in brain function, learning, and memory.
And finally, a truly unconventional class of messengers, gases, specifically nitric oxide, NO, and carbon monoxide, CO. How do these simple gases act as chemical signals in the brain? It seems counterintuitive.
It does seem counterintuitive, but they are indeed important signaling molecules. Both constitutive, always active, and inducible, activated under certain conditions, like inflammation, forms of nitric oxide synthase, NOS, are expressed in the brain, producing NO gas. The application of NOS inhibitors, which block NO production, and NO donors, which release NO, suggests NO's involvement in a host of CNS phenomena, including long-term potentiation, plasticity, activation of soluble guanylyl cyclase, a key intracellular signaling enzyme, modulation of neurotransmitter release, and even enhancement of glutamate-mediated neurotoxicity under certain pathological conditions. Carbon monoxide, another diffusible gas, is generated in specific neurons or glia by enzymes called heme oxygenase. It may act as an intracellular messenger stimulating soluble guanylyl cyclase through non-synaptic actions, meaning it can diffuse freely across membranes and affect nearby cells without needing to be released at a specific synapse in the traditional way. This further demonstrates the astonishing diversity and ingenuity of the brain's communication methods. 6. The signal's end, termination of neurotransmitter action.
Okay, so we've explored how signals are generated, transmitted, and modulated by this incredible array of messengers. But it's just as essential that there are precise mechanisms to terminate the actions of released neurotransmitters. This is absolutely crucial, right? For maintaining the delicate balance of neuronal signaling and preventing constant, uncontrolled activation. What are the primary mechanisms the brain uses to bring these chemical dialogues to a neat close?
Yes, termination is absolutely critical for proper function. There are two main, highly efficient ways the brain ensures precise termination of most classical neurotransmitter signals. One is the enzymatic inactivation of the transmitter. This means its conversion into an inactive compound via a specific enzymatic reaction, often right there in the synaptic cleft. The best and most classic example of this is acetylcholine. Its action is terminated almost instantly via hydrolysis by the enzyme acetylcholinesterase, breaking it down into choline and acetate, which are then recycled. The second major mechanism involves the rapid clearance of the neurotransmitter by specialized transport proteins. These are embedded in neuronal or glial membranes surrounding the synapse. These transporters act like sophisticated vacuum cleaners, swiftly removing the transmitter from the extracellular space, thus preventing it from continuing to act on target receptors. The reuptake of dopamine by the dopamine transporter, DAT, is a prime example of this strategy. In addition to these primary mechanisms, slower processes like simple diffusion of the transmitter away from the synapse and subsequent degradation also play a role, particularly for larger molecules like neuropeptides, which don't typically have dedicated rapid reuptake systems.
Let's delve into these specific transport systems a bit more, as they're often major drug targets. For monoamines, GABA, and glycine, the research highlights specific plasma membrane transporters found on neurons and glial cells.
These transporters are absolutely critical for precise signaling in these systems. For monoamines, we have the norepinephrine transporter, NET, the serotonin transporter, SERT, and the dopamine transporter, DAT. These proteins effectively remove norepinephrine, serotonin, and dopamine from the extracellular space by transporting them back into the presynaptic neuron, or sometimes glial cells. This is a major, if not the primary mechanism for limiting the extent and duration of neurotransmitter signaling in these monoamine systems. To accomplish this energetically unfavorable task, moving transmitters against their concentration gradient, they couple the movement of neurotransmitters to the influx of multiple sodium ions, essentially using the strong electrochemical gradient for sodium as the energy source for inward transport. Structurally, these carriers for Ne, 5-HT, DA, GABA, and glycine typically share a common architecture. usually 12 hydrophobic membrane-spanning domains with their amino and carboxyl termini located within the cytoplasm. They are generally glycosylated within an extracellular loop and possess specific sites for phosphorylation and binding to intracellular regulatory proteins, primarily on their amino and carboxyl termini. This complex structure allows for precise regulation of their activity by the cell.
And then there's a second distinct family of plasma membrane neurotransmitter transporters that specifically handle glutamate and aspartate, called EATs, or excitatory amino acid transporters.
These are equally vital, especially given glutamate's potential for excitotoxicity. There are 5 subtypes in humans, EAT1 through EAT5. They mediate the efficient clearance of glutamate and aspartate released during synaptic transmission. Crucially, the 2 main glial carriers, EAT1 and EAT2, are responsible for the bulk of glutamate transport activity in the CNS. Their rapid and efficient removal of glutamate is absolutely critical for limiting the excitotoxic actions of glutamate that we discussed earlier. They prevent runaway excitation and potential neuronal damage. Structurally, these transporters assemble as home trimmers within the membrane. 3 identical protein units come together to form the functional transporter. Each protomer or subunit possesses 8 transmembrane domains in 2 reentrant hairpin loops, HP1 and HP2, that appear to serve as intracellular and extracellular gates during the transport process, guiding the neurotransmitter across the membrane.
And finally, we have vesicular neurotransmitter transporters. These are fascinating because they don't just clear neurotransmitters from the synapse, they sequester them within synaptic vesicles, packaging them up, preparing them for future release.
Yes, these are indispensable for allowing rapid, repetitive signaling to occur. The research indicates there are at least 3 distinct gene families for these transporters that sequester the neurotransmitters within synaptic vesicles for storage and ultimately for release. These include the VMAT family, VMAT1 and VMAT2 for monoamines, and the related vesicular acetylcholine transporter, VHEST, for HE. There's also VGAT for both GABA and glycine. Interestingly, the same transporter handles both. And 3 distinct vesicular glutamate carriers, VGLUT1, VGLUT2, and VGLUT3 for packaging glutamate. Their purpose is essentially twofold. They ensure that vesicles fill rapidly during neurotransmission, allowing for sustained release when needed, and they provide a means for reducing cytoplasmic concentrations of neurotransmitters in nerve terminals, especially where rates of reuptake are high. This prevents buildup and potential toxicity within the neuron itself.
And this has direct and powerful clinical application, doesn't it? The research mentions that structurally related VMAT2 inhibitors like tetrabenazine, tetrabenazine, and valbenazine effectively lower the vesicular storage and subsequent release of dopamine, and they are now used clinically in the treatment of involuntary movement disorders such as Huntington's chorea and tardive dyskinesia.
They are indeed vital therapeutic tools for those specific conditions. And the plasma membrane monoamine transporters, DAT, NET, and SERT, are perhaps even more widely known as well-established targets for therapeutic antidepressants and for addictive drugs, including cocaine and amphetamines. The core principle here is that selective inhibitors of these carriers can increase the duration and spatial extent of the actions of neurotransmitters in the synaptic cleft simply by preventing their rapid removal. For example, inhibitors of the uptake of norepinephrine and serotonin, SNRIs, are widely used to treat depression and other behavioral disorders by increasing the availability of these mood-regulating neurotransmitters at the synapse. And a fascinating paradox emerges with psychostimulants like methylphenidate and amphetamine, the major drugs used to treat ADHD. While these drugs have stimulant actions in healthy individuals, in ADHD patients, they paradoxically reduce hyperactivity and increase attention. They achieve this primarily by inhibiting DAT and NET, thereby enhancing dopamine and norepinephrine neurotransmission in specific brain circuits thought to be involved in attention and impulse control, ultimately improving focus.
So what does this all mean for us, the listeners? From a medical perspective, these transporters are like the bouncers at the brain's chemical club, right? Meticulously controlling who gets in and out of the synapse and for how long. And by subtly influencing them, we can profoundly shift brain chemistry, either to treat debilitating diseases or, unfortunately, to contribute to addiction. It's all about precisely controlling the signal's end, and it's a testament to the power of understanding these molecular gatekeepers. 7. Forging the future. CNS drug discovery and development. Okay, so having mapped out the brain's complex chemical language from its cellular architecture to the intricate dance of neurotransmitters and their termination, the next monumental step in advancing human health is to take all this knowledge and actually apply it. After identifying a CNS target where modulation would be beneficial for treating a disorder, the immense quest then begins. quote, the discovery of a compound suitable for targeting that site that can be developed into a drug. The research outlines 4 core, often complementary approaches typically used for this quest.
Right. The first approach mentioned involves the synthesis of structural analogs of known neurotransmitters or known modulators of specific receptors and ion channels. The goal here is to systematically generate structure-activity relationship, R information. This involves meticulously determining how even slight changes in a chemical structure can lead to greater target specificity, enhanced potency, or improved safety profiles. It's essentially about refining existing chemical keys to fit the biological locks even better, making them more effective or safer.
Then there's the incredibly diverse approach involving the use of natural products. These are compounds that exist naturally, maybe toxins from plants or animals, or extracts from medicinal herbs that have shown some efficacy for treating a particular disease historically or in screening. These can be used either as drugs themselves, if they happen to have the right properties, or perhaps more often as direct chemical templates for designing entirely new synthetic drugs with improved characteristics like better stability or oral availability.
A third core approach, which has really revolutionized drug discovery over the last few decades, is the development of a signaling assay that is amenable for the screening of large compound libraries. This is high-throughput screening, or HTS. It allows researchers to rapidly test thousands, sometimes even millions, of diverse chemical structures in parallel against a specific biological target in an automated fashion. The aim is to quickly identify novel scaffolds with promising functional profiles, completely new chemical starting points that can then be chemically optimized for drug development.
And the fourth approach, which brings us full circle to what we discussed earlier with structural biology, is the cutting-edge use of atomic-level structures of targets. These structures are obtained through sophisticated techniques like X-ray crystallography or cryo-electron microscopy.
Exactly. Using these incredibly precise atomic blueprints of the target protein, scientists can then rationally design new compounds with a much higher degree of predictability about how they might bind and interact. Or they can employ structure-based virtual in silico screening. This means using powerful computer models and algorithms to sift through vast digital libraries of chemical structures to identify chemical scaffolds that likely bind to the target with high affinity and specificity based on the known 3D structure. Each of these 4 approaches has its own benefits and potential pitfalls, of course. But when used together, often in a synergistic manner, these types of approaches have been remarkably successful in discovering and developing many groundbreaking new drugs for targeting CNS signaling processes. They're really pushing the boundaries of what's medically possible. What's truly fascinating here is the evolution of these methods. We've gone from early, almost trial and error work with natural compounds to highly sophisticated, computer-aided design based on these detailed molecular blueprints. This convergence of diverse scientific disciplines, chemistry, biology, physics, computer science, is truly the frontier of neuroscience drug discovery, allowing for unprecedented precision and speed in therapeutic development. Outro?
And that brings us to the culmination of our deep dive today into the brain's astonishing chemical language. We've journeyed from the basic cellular architecture of the brain through the incredible electrical and chemical dances of neurons. We met the diverse cast of neurotransmitters, from the fast-acting amino acids to the subtle, long-lasting peptides and purines, and even gases. And we explored the sophisticated mechanisms that precisely terminate their actions, which are just as important as the signals themselves. We've truly seen how profound and intricate the understanding of this field is, and why mastering it, as our expert mentioned, is so critical for advancing human health through groundbreaking drug development.
Absolutely. This comprehensive deep dive into the CNS reveals not just the extraordinary complexity and, frankly, elegance of the brain, but also the immense, still unfolding potential to understand and intervene when things go awry. Consider this as a final thought. As we continue to uncover more about the subtle interplay of these myriad chemical messengers, and as our tools for mapping and modulating them become ever more precise, what entirely new frontiers of human experience, things like the very nature of consciousness or the nuances of empathy and creativity, might we begin to decode and perhaps even gently, ethically modulate in the years to come? It's a profound question.
An incredible and provocative thought to leave you with indeed. We encourage you, our listeners, to continue your own deep dive, perhaps by reflecting on how the information we've shared today applies to conditions you know of, or maybe just appreciating the silent, complex, and truly astonishing symphony happening inside your own head right now. Until next time, keep exploring, keep questioning, and keep being well informed.